All right, class, today we're going to talk about vesicular traffic, secretion, and endocytosis. Okay, so let's look at the secretory and the endocytic pathways of protein sorting. And so the secretory and endocytic pathway protein trafficking, there's this kind of unifying principle where transport vesicles transport membrane and soluble proteins from one membrane-bound compartment to another. And so these transport vesicles collect cargo, which is proteins, in the membrane that bud from a donor compartment. And they deliver cargo proteins to the next compartment by fusing with that target membrane. So you can kind of think of them as like a bubble forming off of a membrane that captures some of those cargo proteins and then are transported to another membrane, and that bubble kind of merges with the next membrane. So in the secretory pathway, the distribution of soluble and membrane proteins is synthesized by the rough ER, which we talked about in the previous chapter. And the final destinations are the cell surface or the lysosomes. And this occurs in two stages. In stage one is what we talked about, I believe it was last chapter, where synthesis of proteins that are bearing this ER signal or targeting sequence. There's co-translational insertion into the ER of these newly made polypeptide chains, and those get inserted into either the ER membrane or across it into the ER lumen. And so we talked about how the ribosomes bind to those anchors on the membrane of the ER, and they're actually transcribed into the ER. So that's stage one. And stage two is the actual trafficking of these proteins. And so we'll start, so we already talked about this kind of last chapter, so we're going to start with number 2 right over here. And so proteins are packaged into these transport vesicles that bud from the ER and fuse together to form this new cis-Golgi cisternae. So the Golgi apparatus is these kind of layers of membranes, as you can see here, and these stacks can be up to 100 or so stacks of them, depending on the organism. It is different across all eukaryotes. But the cis side is the side closest to the ER, and the trans side is the side furthest from the ER. And each one of these is a cisterna, or cisternae for plural. Now, so the vesicle here, as you can see, it will fuse with this Golgi cisternae, and it will dump its enzymes into that cisternae. And in step 3, we have retrograde Golgi to ER transport. So retrograde means kind of like in the opposite direction. And so there's some proteins that are ER transport specific, the proteins that are responsible for this process over here on the left that we want to reuse, right? So the ones that are kind of targeting the cargo and helping the cargo get to where it needs to go. Well, once it gets to the Golgi, those can be sent back to the ER to be used for the next transport. So there is a step of retrograde signaling there. And then in step 4, we have this cis-Golgi cisternae and its contents are moved from the cis to the trans face of the Golgi complex by non-vesicular cisternal maturation. And so what that's saying is the cisternae will move themselves and their contents towards the trans face, and in the process of being within the Golgi, we're doing protein modification. So the reason that we're even in the Golgi, besides targeting and move it to where they need to go, the reason we go through all these layers is because a lot of the post-translational modifications, including adding those mannose sugars and removing different functional groups and things of that of these proteins, is done within this Golgi apparatus, or adding targeting sequences, etc. So once they get to the trans-Golgi side here, we have, also, before I get to that step 5, I don't want to jump too far, there's also retrograde transport from a later cisterna to an earlier one. And this is those Golgi-specific enzymes that are required for those modifications. So we don't want them to get excreted out of the cell because they'll go out there and have nothing to do. We want to recycle those, right? Those are only there to target or to modify the proteins within the Golgi. So we want to recycle that. So we dump those back into the previous cisterna that can then use it to modify its proteins. So these are continually recycled. So everything on this recycle, the retrograde side over on the right here, those are all things that are enzymes or proteins specific to the Golgi or the ER. They're not meant to be trafficked. The trafficked proteins, they don't go retrograde. Okay, so step 6 here is the constitutive secretion. So once we've got this trans-Golgi network up here, all of our proteins are furthest away from the ER, they're ready to be excreted, and we have constitutive secretion, and this is where we have secretion of soluble proteins and also membrane proteins. So if you're a protein that is stuck in the membrane, like this one right here, when you become a vesicle, you're still stuck in the membrane, and then when you bind to the plasma membrane of the cell, you're still stuck in the membrane. So it preserves them being in the membrane. And if you are a soluble protein, not in the membrane, you get excreted out of the cell here. And constitutive means this is always on. So these are proteins that are always excreted. In step 7, there's regulated secretion. So these are proteins that are secreted only from certain cell types or at only specific times, like digestive enzymes. Your skin cells don't secrete digestive enzymes. Well, constitutive secretion is all your cells do it all the time. So regulated cell types, digestive enzymes, insulin, things like that. That also happens, and that requires there's a regulation, molecular regulation, which we're not going to talk about right now. And so let's see, step 8 now. Some of these are sorted to the lysosome, which we'll get into a little bit more. And then in step 9, we have the endocytic pathway, and this is where vesicles budding from the plasma membrane take up soluble extracellular proteins and deliver them to the lysosome for via late endosomes. And so we can also kind of go in the opposite direction, where we take things from the outside and deliver them to the lysosomes, and things from the inside can be delivered to the lysosomes. And like I said, we'll get into that a little bit later. So let's talk about how these vesicles bud and fuse with the target membrane. And so vesicles bud from a donor membrane, and they fuse with a specific target membrane. An assembly of a protein coat drives vesicle formation and the selection of specific cargo molecules. So we're going to get into that here. In part A, we see that recruitment of these GTP binding G proteins to a region of the donor membrane is what kind of starts this. And as you can see, there are these blue circles right here. And cytosolic coat protein complexes bind to the cytosolic domain of these membrane cargo proteins. And there's also membrane cargo receptor proteins as well. And so that's where the coat proteins bind. They're transmembrane proteins, so they'll bind to the cytosolic domain. Some cargo proteins act as receptors to bind soluble proteins in the lumen, so that's what these membrane cargo receptor proteins do. So they aid in attaching to the non-membrane bound proteins that need to be trafficked, the cargo that isn't in the membrane. In the donor membrane, there are these specific snare proteins, or here it's V-snare for vesicle snare, that are captured in the budding vesicle membrane. And these snare proteins are what target that vesicle to where it needs to go. And so once the coat proteins, as you can see, they're kind of shown to be like semicircles or curved. And when they are concave, I guess would be a good way to put it. When they bind to this membrane, they form it to their shape and kind of pinch it off, right? And so they end up closing off this vesicle. And then once that vesicle gets pinched off, in the cytosol, those coat proteins are then removed. So then on the outermost layer, we have these snare proteins exposed. And so those snare proteins are what target the vesicle to a specific membrane, whether it be the lysosome or the cell membrane or whatever. And so these snare proteins will interact with T snare proteins of the target, so that's target snare proteins, which will start the process of the fusion of the two membranes. So here is a microscope image of this budding process. So you can see the beginning of the formation, you can see the coating of those coat proteins, and you can see that it's starting to pinch off in both of these two vesicles here. And so just kind of put an image to the theory that we just talked about. Okay, so now let's talk about how these proteins assemble, that coat assemble on a vesicle, and the role of SAR1 in this assembly. And so in our first step here, SAR-GDP, which is right up here, that interacts with this recruitment protein on the ER membrane called Sec12. And this is an integral membrane protein that is transmembrane and found throughout the ER lumen. We then have Sec12 kind of hold SAR1 in place and stimulate this exchange of GTP in and GDP out. So when GTP enters SAR1, it changes the conformation and it causes the formation of this hydrophobic N-terminus. And since that forms while really close to the membrane, that hydrophobic N-terminus, to get away from water, inserts itself into the membrane. So now we've created a SAR1 membrane protein. In step 2, SAR1 GTP, which is now what we have here, it recruits Sec23 and Sec24, which are these proteins. So as you can see here, this complex of Sec23, Sec24 has specific domains that are specific to bind to SAR1 and this transmembrane protein here. And this Sec21, the activity of this, this Sec23, 24, bind specific short sequences of these transmembrane cargo proteins. So they are specific for the cargo sequence, the cargo proteins that are going to be transported. And the coat assembly, as I said in the previous slides, because these are curved proteins, when they force this bind, they kind of force this circle to be made because they're concave like this, right? And so they're pulling the plasma membrane into the circle, which will eventually pinch off the plasma membrane into a vesicle. So once we're in this vesicle formation, what happens is this Sec23 that is on the outside, this green coat protein here, that re-stimulates the hydrolysis of the GTP in SAR1 to go back to GDP and a phosphate group, right? And when that occurs, we lose, let me get a different color here, we lose this hydrophobic domain that we created in the first place, and so that causes this SAR1 protein to no longer have a hydrophobic tail and kind of be shot out of the nice hydrophobic membrane that it was hiding in. And when it does that, it also releases all those coat proteins, because the only thing those coat proteins are bound to very rigidly is SAR1. They are bound a little bit to these transmembrane proteins, but not enough to hold it in place when there's this conformation change. And so this all falls apart. And so that's when the vesicle makes it into the cytosol, we lose the coat proteins, and we lose SAR1, which was there to help in the formation of those coat proteins. And what we're left with were these target proteins that will be able to guide the protein, the V-snare proteins. They'll be able to guide it to where it needs to go. So as you can see, we had one example of the V-snare protein, but there are a lot of different places proteins can go, right? So they can go back to the nucleus, they can be excreted, they can go to the lysosome, they go to the Golgi, they go to chloroplast in plants, etc. So there's a lot of trafficking and decisions that need to be made where these proteins go. So because of that, there's a lot of different signal sequences and proteins, etc., that are associated with this. And so as you can see here, the different signal-bearing proteins and proteins with the signal. So I don't want you to memorize these, but just know that there are a lot of different signal proteins, because there's a lot of different places that proteins need to be trafficked to and from. Okay, so we've talked about how these proteins are packaged into cargo into vesicles, and we talked about how they get sent different places, but now how do they bind with their target membrane? So let's keep working through this process, right? So this is the model for docking and fusion of these transport vesicles with their target membranes. So RAB GTPases control docking of vesicles on the target membrane. So you can see RAB right here. And so it starts with fusion of these secretory vesicles with the plasma membrane. And similar mechanisms kind of mediate all vesicle fusion events. There's slight differences, but for the most part, they're very similar. So in step one, which is right here, we have the transport vesicle docks on the appropriate target membrane. And that RAB protein that I had just mentioned, that is tethered via this lipid anchor to this secretory vesicle, and it binds to an effector protein complex on the target membrane. So as you can see, there's kind of this binding lipid anchor, and then it binds to this RAB effector, which they have is kind of this light blue kind of square-ish or rectangle-ish with an indent for binding to RAB on the receptor membrane there. So in step two, that V-snare protein forms stable coiled coil interactions with these T-snare cytosolic domains. So remember, there is this V-snare, which is the vesicle snare protein or targeting protein, and T-snare, which is the receptor protein. And so they form this coil-coil interaction, which you can kind of see they like wrap around each other. It kind of looks like licorice or something, or candy cane. And so once that happens, these snare complexes hold the vesicle close to the target membrane. So they're there to make sure that they put everything in the right proximity. And so remember, they're specific for where we're targeting to, right? And so if we're going to the lysosome, we're going to have different of these snare proteins compared to if we're going to excrete out of the membrane. And so in step 3 here then, we have fusion of those two membranes. And so this drives the dissociation of the snare complex when the membranes freeze, and that frees the snare proteins for another round of vesicle fusion. And so they can be reused in that case. And at this point, RAB-GTP hydrolyzes to RAB-GDP and dissociates from the RAB effector, which isn't shown. So this was still in place at this point, and when the membrane binds down here, that frees RAB to leave. So RAB is out of here. All right, and then once that happens, we have, we're dumping all of our cargo into whatever vesicle or membrane or lysosome that we're attaching to, and we've completely moved our target here. And B here, we're just showing the kind of ribbon of the V snare, T snare complex. So V snare, obviously the vesicles snare protein, and then T snare, which is the target receptor protein. And as you can see, this is kind of how they intertwine. We have these long alpha helices that wrap around each other. And my bad, it looks like licorice in the previous one. I don't know what it looks like here, rope, I guess. But yes, so that's how they bind and hold everything in proximity for the merging of these two membranes. You can tell it's been a long semester. I'm looking for things to make analogies for. Bear with me, we're almost done here. Okay, so let's look at specifically at the vesicle-mediated transport of protein between the ER and the cis Golgi. Remember, the cis Golgi is the closest part of the Golgi apparatus. And so the transport between the ER and the cis Golgi is the initial transport stage of this secretory pathway. And the forward, not retrograde, but anterograde transport starts with this COP2 vesicle-mediated ER to cis Golgi transport, which we'll get into a little more in a second here. And so the cargo of this is indicated here. These are newly synthesized soluble proteins. And in step 1, the COP2 coat assembles on the ER membrane to form vesicles which contain those snares that we talked about, the trafficking snares, which are orange there, the membrane cargo proteins, which are blue, the soluble cargo proteins, which are magenta, and they're bound to the appropriate receptors for them in this budding membrane vesicle. And then the COP2-proteid vesicle pinches off to be freed of the ER. And then in step 2, that COP2 coat disassembles and exposes those V snare proteins to the vesicle surface for the fusion target, right? So these green things, they take off, those are the coat proteins, and that exposes those orange snare proteins that we had embedded in the membrane. In step 3, the RAB effector interaction promotes association of those V snares with the T snares on the cis Golgi membrane. So that was what we talked about in the last slide, where we formed that what I said was Twizzlers, but then when we looked at the ribbon structure, it didn't look as much like Twizzlers as I had thought. But it wraps around the target snare and the vesicle snare. And then the membrane fusion releases those vesicle constituents into the receptive Golgi. So we release everything in there into the compartment of this membrane. Now, in the reverse or retrograde transport, those COP1 vesicle-mediated cis Golgi transports to the back to the ER. So we have COP1 instead of COP2. And the cargo is, this recycles part of the membrane bilayer So we're recycling some of those target proteins, right? The V snares, and as well as missorted ER proteins and any modification proteins. So we're not showing them, but there'll be proteins in here that went to the wrong spot, etc. Those can be recycled back to the ER. So then in step 4 here, we are recruiting this COP1 coat to target it back to the ER. And in step 5, this COP1 coated vesicle pinches off the cis-Golgi membrane and is free. And then in step 6, the cis-Golgi snare interaction binds again, just like it does with the Golgi network going the other way in the anterograde. In the retrograde, it binds with the ERT snares, which mediates vesicle fusion to deliver the cargo back to the ER. So it's kind of this circle or cycle that's happening between the ER and the Golgi. So we talked about this retrograde trafficking from the Golgi back to the ER. So let's kind of look at how this works. So there are proteins that sometimes get sucked into these vesicles, and they're not supposed to be there. So these are soluble proteins that are supposed to be down here in the ER. And these proteins are actually enzymes that, like I said before, they modify newly synthesized proteins. And there are several types, and they contain this C-terminal K-del. It's on here somewhere. Oh, here we go. K-del receptor, or K-del peptide, and there are receptors for this peptide too. So they contain this K-del peptide, and sometimes, like I said, they get missorted into these vesicles that then end up in the Golgi network. And so what happens is these peptides, they will bind to these K-del receptors once they get to the Golgi network. And when they do this, these K-del receptors prevent them from being sent elsewhere. And instead, those K-del receptors are incorporated into this COP1 or COP1 coat. This COP1 coated vesicle, and not just the coat, but the COP1 coated vesicle, and they are returned then to the ER. And so, as you can see too, it's important to know that these K-del receptors are kind of recycled in a cyclical manner too. So this membrane will fuse to the ER membrane, and this membrane-bound K-del receptor will continue to be in the cell membrane of the ER. And as you can see over in step 1, this K-del receptor is incorporated in the vesicle when it goes to the Golgi, and then it kind of pulls back any of those enzymes that are modifying proteins and returns them to the ER. Okay, so we've talked about oligosaccharides and how they are added to proteins, and this can be for anchoring or a whole bunch of different reasons, but one of the reasons was for targeting. And so it would make sense that in this vesicle system that proteins would be, their oligosaccharides would be modified for this purpose. And so we're going to talk about how they're modified in vertebrate cells here. And we have 3 layers of the Golgi. We have the cis and the trans that we had talked about previously. And then the in-between layers, we call them medial. And it depends on the organism how many layers there are. But the middle is just medial. So this happens in the anterograde transport, so going forward and not back to the ER. And there are specific transferase enzymes that add sugars to these oligosaccharides from sugar nucleotide precursors that are taken in from the cytosol. And so in this process here, we start with the cis-Golgi, and we start by removing 3 mannose sugars. So this one and these 2 are removed from the oligosaccharide. And at that step then, the protein is moved to the medial portion of the Golgi apparatus. And in this step, we are adding a 3N-acetylglucosamine, and we are removing 2 more of those mannoses, so they're out of there. And then we add at the end a single fructose, which is that. From there, we move to the trans Golgi, where we add 3 galactose, which are the orange dots here, and we add a N-acetylneurominic, excuse me. It's getting late. As you can see, the lights have changed in the room because it is late, so I'm stumbling a little bit here. So N-acetylneurominic acid residue is added to each of the galactose residues. And so that is these purple that are added on top of the galactose. Okay, now moving from the trans Golgi network, how do things get to where they're going from here? So continuing down our line, there in the trans Golgi network, there is a distal sorting compartment, and this sorts proteins into 5 different types of vesicles for transportation to the plasma membrane, the endosomes, or the lysosomes. And so there are 5 destinations, hence the 5 different types of vesicles. There are the COP1 vesicles, which are for retrograde transport, which are this guy here to go back to the trans Golgi or further down towards the ER. There are AP complex vesicles that potentially have a clathrin coat, and these transport to the lysosomal or to the lysosome, and they transport lysosomal enzymes directly to that lysosome. So that is to here. So they're AP complex, but like as I said, they could potentially have that clathrin coat. And then we have the clathrin coated AP2 vesicles that transport the lysosomal enzymes to late endosomes for eventual delivery to the lysosome. So they are kind of temporary holding before being transferred to the lysosome. And 4, we have the constitutive secretory vesicles, which we don't know much about their coat, at least at the time of this publication or of this textbook being made. And these are the ones that, as we said earlier, they're secreted relatively constantly, and they're secreted by all cells. And then we have, lastly, the regulated vesicles, and these are, as our example was, things like specialized cells that secrete only some of the time. And this could be digestive enzymes, peptide hormones, insulin, things like that, where special cells make them to secrete them into, say, the blood cardiovascular system, the bloodstream, to adrenaline and things of that nature. So as you can see here, we have our clathrin coat. It's this fibrous coat that goes around the vesicle. We have our cargo protein inside with the receptor protein, and we have the integrated cargo protein. We have our AP complex of these coat proteins. What's interesting though with these clathrin coated vesicles is that there is a protein called dynamin, which is required for pinching off these vesicles. And it polymerizes around the neck of the vesicle, as you can see here, and it hydrolyzes GTP to GDP and causes a conformational change, which is coupled with that membrane fusion, and it releases the vesicle from the donor membrane. And this is something that's specific to this type of vesicle. So we talked about the lysosome a couple slides ago, specifically how the AP complex or the clathrin coating will get a vesicle to the lysosome. And that delivers these lysosomal enzymes. But how do we know what enzymes are lysosomal enzymes, and how do we get them into those vesicles that are going to the lysosome? So just like before with oligosaccharides, we add a tag to them, an oligosaccharide tag that tells us how to get there. And so the way that this works is for enzymes going to the lysosome, soluble enzymes, so ones that aren't in the membrane, we tag them with mannose 6-phosphate. And the way this works is when a lysosomal enzyme is synthesized, a M6P or mannose 6-phosphate residue is added. And this is done by two cis-Golgi resident enzymes. So these are enzymes in the cis-Golgi part of the Golgi apparatus. And so in step 1, there is this protein here, or enzyme, which is a protein, which is cis-Golgi localized N-acetylglucosamine phosphotransferase. So very long name here. And what that does is it recognizes the specific sequence in a newly synthesized lysosome enzyme. So here you can see there's kind of 3 parts of this folded peptide chain that are recognition sequences. And it transfers a phosphorylated GLC NAC, we'll call it. So you're not going to be forced to remember cis-Golgi localized N-acetylglucosamine phosphotransferase. So GLC NAC-C, or GLC NAC, excuse me. It'll transfer a phosphorylated one to carbon-6 of one or more of these mannose residues. Remember, so we have this kind of mannose chain here that can get modified. So it's adding this phosphorylated group to carbon-6 of this mannose residue. And then phosphodiestertransferase transferase removes that the GLC NAC group, leaving only the 6-phosphorylated mannose residue on this enzyme. So it removes the GLC NAC, and we just have a phosphorylated mannose, and that 6-phosphorylated mannose is then targeted to the vesicles that will end up at the lysosome. Okay, let's continue to look at how these are trafficked to the lysosome. So we've got our M6P created or tagged protein. So in this, I'm going to start down here at the bottom in step 1. So we have these clathrin coated buds, and this M6P receptor in the membrane recruit M6P lysosomal enzymes into this clathrin or AP1 coated vesicle. So as stated before, we've tagged it, so now this vesicle will look for that mannose 6-phosphate, and that's what that little kind of this orange sickle here is to bind those lysosomal enzymes. And then in step 2, the vesicle, well, it's released first into this vesicle, then the coating is removed, just like in our previous talks of vesicles making it into the cytosol. But in step 2A here, we have that these coat proteins are then recycled. So the clathrin and the AP complex are then brought back for the next vesicle. So they're just there to kind of help in the budding process. In step 3, over here, we have fusing with the late endosome compartment, and this acidic endosome pH dissociates this M6P enzyme from the receptor. So because there's an increase or a lower, let's go make the arrow down here, pH in the endosome, we are going to release the protein from our receptor and the phosphate group from the protein. So step 4A over here, we have the M6P receptors are recycled and captured in vesicles that are targeted back to the Golgi. So really anything that isn't the cargo is recycled. It's a very efficient system, right? Because we don't need to keep making these M6P receptors or these coat proteins if they're just there for a certain time. So they're recycled just like the coat proteins are. Step 4 here, we have the endosome that is combines with the lysosome, and we have delivered our enzyme to its final destination. However, there are modifications to this. So step 5 over here, we see that some receptors are actually delivered to the cell surface after this process. So this process can also take enzymes from the exterior of the cell and deliver them to the lysosome, not just from the Golgi network. So we're going to look into this a little bit. So step 6 at the top here. So this receptor is now on the cell surface. And so in step 6, the plasma membrane M6P receptor can capture these enzymes and bring them in for the endocytosis pathway. And the coat will be in place just like before. So we have a clathrin-coated pit, as they say. So in endocytosis, that invagination we call the pit. And we have this vesicle, which similarly to step 2, will lose its coating. And in step 8, will, just as before, fuse with the late endosome and dissociate from the receptor, or the protein will dissociate from the receptor for delivery to the lysosome. So we can either come from the Golgi or we come from the exterior of the cell to the lysosome. So let's take a look here at some low-density lipoprotein. And these are important because cells take up lipids from the blood in the form of large, well-defined lipoprotein complexes. And all of these have a very similar structure, the same general structure. They have a shell that is composed of apolipoprotein and a phospholipid monolayer, not a bilayer. Important to note that it is a monolayer, so you can see the polar heads and the nonpolar chains, but there is no corresponding second layer. And they also have a hydrophobic core that's mostly composed of cholesterol esters or triglycerides, but there's sometimes a minor amount of other neutral lipids, such as vitamins, that can be inside them. And the LDL particle contains only a single molecule of one type of apolipoprotein wrapped around the outside of the particle. So as you can see, this big apolipoprotein B wrapped around the side. So we said that LDL is brought into the cell through endocytosis, so let's look at this mechanism. So we start with the cell surface LDL receptor, which is looking for those LDL particles. And there is an extracellular domain on it that binds this ApoB protein, or this surface protein, tightly at this normal pH. As long as the blood pH is normal, then it will bind it. The cytosolic tail MPXY sorting signal it interacts with this AP2 complex and incorporates the LDL receptor complex into the clathrin-coated pit. And so once in the pit, the clathrin-coated pit-containing complex pinches off as the vesicle is interacting with dynamin again, right? So that dynamin mechanism pinches off this vesicle. It moves into the cytoplasm, and just like in all of our other examples, the coat then leaves. So we have no more coating. In step 3, the vesicle fuses with the endosome, and just like previously, with our last example of the lysosome, the acidic pH of the endosome causes a conformational change in the receptor, which releases that LDL particle. So you can see the pH is real low here, not close to neutral. In step 5, the LDL receptor is recycled to the cell surface. So this is, same story, but different trafficking. So why reinvent the wheel if it works for trafficking to the lysosome? So we're going to save that LDL receptor back to the cell surface. And in step 4, the actual LDL, it fuses with the lysosome, and the LDL particle is broken down into those constituent parts that we talked about in the last slide by these lysosomal enzymes. And so we have fatty acids, cholesterol, amino acids, etc., vitamins if they're in there, and then these are recovered by the cell for nutrients, for metabolism. Okay, so let's look at how cells uptake iron. And I'm sure there'll be some similar mechanisms here compared to the rest of our uptake and transfer pathways. So we're going to look at transferrin. So transferrin is a protein that carries iron, and there's apotransferrin, and apotransferrin is when there's no bound iron, and then we have ferrotransferrin that we're going to start with that has bound iron, and this is in the blood. And so in this mechanism, this ferrotransferrin, it's a dimer, so it'll carry 2 iron molecules, and it binds tightly to this cell receptor at neutral pH. And this is a transferrin-specific receptor. The transferrin receptor, the cytoplasmic tail, it interacts with an AP2 adapter complex, which incorporates the receptor and the bound ferrotransferrin into this endocytic clathrin-coated vesicle. So not really shown here, but there's interactions with this tail domain, and the AP2 adapter complex, which are these blue coats, will come and bind to this guy, and that will start the process of making this vesicle. So once inside, just like before, we lose our coating, just like all of our other examples, and just like our other examples, we also recycle our coating, the proteins on the coat, for the next vesicle. In step 4, we have fusion of the vesicle to the late endosome. And at this point, where things are a little different here, the ferrotransferrin and transferrin receptor never leave the membrane. So once we get to the late endosome, the pH will cause the release of iron from the ferrotransferrin, and it's reduced from iron 3 to iron 2 by this endosome reductase, and it's transported into the cytoplasm here. Now, as said before, our enzyme never leaves the receptor, which is also bound to the membrane. So at this point, it's called apotransferrin because it doesn't have the iron, so there's no ferro anymore, right? And so the apotransferrin is then recycled to the cell surface, and as you can see, as we're getting to the cell surface, our pH is increasing, and once we get to neutral pH up here at the cell surface, the apotransferrin dissociates, and then it can go and recruit more iron to be bound and then chaperone it back into the cell. So how do we break down proteins that are in the plasma membrane? Because they get damaged just like any other protein, eventually need to be broken down. So the way that this works is they're going to be delivered to the lysosome, just like other proteins and things that need to be broken down. And the mechanism for this is that we have these newly synthesized lysosomal targeting proteins from the trans Golgi network, and they get packaged into vesicles and end up fusing with the late endosome. So you can see the green ones, oh, that was blue. They look kind of the same on my computer. The green ones throughout here. And at the same time, we have endosomes that carry these plasma membrane proteins that are blue, and they target them to the late endosome as well. And so as you can see, we now have green. I'm going to try to get these colors right. Like I said, my screen, they're kind of very similar because I have a blue reducing nighttime filter so I can sleep at night. So it makes it a little difficult. Anyways, that's neither here nor there. So we have blue and green. The blue ones are the ones that target to the lysosome, and the blue ones are the ones that were the membrane proteins. I hope I said that right. Green ones from Golgi, blue from plasma membrane. So from there, we see that these blue membrane proteins that were on the extracellular membrane, they get budded off into their own internal vesicles, and we get this multivesicular endosome. So we have vesicles within a large endosome, which is a vesicle. So we have vesicles within vesicles. And at this point, all the green targeting proteins, they stay on the outside, and the ones that came from the plasma membrane that needs destroyed are inside in these little internal vesicles. So those targeting proteins are able to help bring this late endosome to the lysosome, and it will release these internalized vesicles into the lysosome that contain those membrane proteins that wanted to be broken down from the plasma membrane, and they will be degraded within here, as well as the internalized vesicles. So how do these multivesicular endosomes form? So as we talked about in a previous lecture, proteins that need to be broken down during stress, or if they're damaged in any way, they get targeted and tagged with ubiquitin, and this is the same for these transmembrane proteins. They get ubiquitin tags put on them, and then at this point, when they get targeted to the early endosome, there is, where are they? Here we go, our cargo proteins that have a ubiquitin tag, and then there are the green proteins, the HRS proteins, that were the target proteins for the targeting to the lysosome. These proteins are also ubiquitinated, and the ubiquitination of the HRS protein directs the loading of those ubiquitinated membrane cargo proteins into the vesicle. So the HRS help get these into the vesicle. In addition, they recruit this escort complex, and the escort complex binds to the ubiquitin tags of those HRS proteins, and it helps to squeeze off this internal vesicle. So it's kind of like that dynamin protein that we talked about earlier. VSP4 or VPS4, excuse me, that will recycle just like everything else within this whole pathway. Everything's recycled, very efficient. It will help remove those escort complex proteins to be used again to pinch off more of these cargo proteins into the internal endosomal vesicle. So the very last thing we're going to look at here in this chapter is the autophagy or autophagic pathway. And this pathway delivers cytosolic proteins and organelles to the lysosome for degradation. And the mechanism for this is that we have these ATG proteins that induce formation of this cup-shaped membrane structure that goes around a portion of the cytosol. So on the right-hand side, we're just surrounding a piece of cytosol, or on the left, an example of a mitochondrion that's kind of being swallowed up by it. And the proteins involved in this include ATG 5, 8, 12, and 16. So as you can see here, we have them listed. And ATG 8 forms the coat around this autophagiosome. And so in step 1, this membrane is continued to be added around and forms an autophagiosome that envelops the contents into 2 complete membranes. So you can see we have 1 and 2 membranes surrounding, in this case, the mitochondrial membrane is also in there. And then in step 2, fusion of the autophagiosome with the outer membrane of the lysosome releases its contents into the lysosome, but it's within a single membrane vesicle, right? Because we had those 2 kind of a bilayer formed before. And so at this point, the vesicle protein and lipid components are degraded by the lysosomal hydrolases, and the amino acids have permease transport across the lysosome membrane into the cytosol for use in protein synthesis. So again, recycling. So all of our amino acids that are broken apart from either our proteins or our proteins that made up the mitochondria, those are exported and to be reused at a later point. So that's the end of this chapter. We're right at an hour, even though we only did 27 slides. As you can see, a lot of these complexes, very, very similar, slight tweakings. But as they say, why invent the wheel, right? So if we're good at trafficking stuff at a high fidelity way, very efficient way, let's just do it for all of them. And that's kind of through evolution what has happened with the cell.